In this video, I'm going to spend some time talking about neurotransmitters. Here I'm going to give you the basics. You're going to pick up a lot more as we go throughout the semester, but this will be a good foundation moving ahead. Neurotransmitters can be divided into families. These families generally are based on their chemical properties and chemical structure. You can see here, first we have amino acids. That's going to include neurotransmitters like glutamate, GABA, glycine, aspartate, and possibly some others. The two that I really want to talk about right now, though, are glutamate and GABA. Glutamate is the major excitatory neurotransmitter in the brain. It's not the only one, but it's the major one. So if we talk about glutamate, we're talking about excitation. GABA is the major inhibitory neurotransmitter. Neurotransmitter in the brain. Okay, so if I tell you something's a GABA neuron, that means when that neuron releases GABA, it's going to quiet other parts down. If it's a glutamate, it's going to excite. Okay, so those, it's important to know they're amino acid neurotransmitters. Part of the reason that it's important to know the family is because of the chemical properties of each family. This will let you know whether it passes through the blood-brain barrier. It lets you know whether it's easily metabolized or not. So there's a lot of things you can learn and know about a neurotransmitter just by knowing which family it's in. There's only one member of this next, so it's not really a family. That's acetylcholine. We'll talk about acetylcholine at the neuromuscular junction. We'll talk a lot of detail about acetylcholine. It's very important for movement. The monoamines sometimes are also, these are derived from amino acids. There's two broad categories. There's the indoleamines and the catecholamines. The only indoleamine we're going to talk about is serotonin. Serotonin is abbreviated 5-HT often. That stands for 5-hydroxytryptamine. And again, that name is based on the chemical structure. You can see why we call it 5-HT or serotonin instead of 5-hydroxytryptamine. The catecholamines are dopamine, which is abbreviated DA, norepinephrine, NE, and epinephrine, E. These, again, are all related. It's important for you to know the indoleamines, catecholamines, monoamines. Later on, we're going to be talking about enzymes. There's something called monoamine oxidase. Monoamine oxidase is going to break down any monoamine. So if I tell you what monoamine oxidase does, you should be able to tell me that that affects serotonin, dopamine, norepinephrine, and epinephrine, but not acetylcholine, glutamate, or GABA. Later on, we're going to talk about neuropeptides. Neuropeptides are either very short proteins or long chains of amino acids. These are going to be important for more complex behavior. We're going to talk about it in the context of pain or feeding. We'll get to that later. Examples of neuropeptides are endorphins, substance P, neuropeptide Y, and many, many other neuropeptides. We'll cover those as we go. These tend to be large. They tend to be built and made in the cell body. Because they're proteins, there's genes for these. Okay, so we'll talk about that more later. Purines like ATP and adenosine. And then there's actually some gas neurotransmitters. The one we're going to talk about is NO, nitric oxide, not nitrous, nitric oxide, NO. This is going to be a retrograde signal. So in general, the flow of information is a presynaptic neuron to the postsynaptic neuron. Nitric oxide is actually going to carry information from the postsynaptic cell back to the presynaptic cell. And we'll talk about its specific role later on in the semester. Now for the neurotransmitters, I want to focus on their synthesis, how they're made. Acetylcholine, dopamine, norepinephrine, epinephrine, and serotonin are all small. There's no gene for these. Okay, there's no gene for acetylcholine. If you want to make acetylcholine, you're going to take acetyl-CoA plus choline. These are chemicals your body makes through metabolism or that you get in your diet. Okay? It's going to take, those two are going to combine together to form acetylcholine. Now that doesn't just happen. If I take a dish of acetyl-CoA and a dish of choline and mix them together, I don't get acetylcholine. That's not a chemical reaction that normally occurs. Instead, an enzyme, choline acetyl transferase. We abbreviate that as CHAT. Choline acetyl transferase is going to take these two components and cause them to bond together in a certain way to form acetylcholine. Okay? So here's something that's important. This is an enzyme. That means it's a large protein. That means there's a gene for it. Okay? So if I want to make more acetylcholine, I can turn on the gene and make more CHAT. The more CHAT I have, the more of this reaction is going to occur and the more acetylcholine I'm going to make. Now, one thing, just sort of as a pet peeve of mine, a lot of times people will talk about choline supplements, that if you take a lot of choline, you'll make more acetylcholine. That's not the case. Unless you're malnourished, you have plenty of choline. Choline isn't what limits how much acetylcholine you have. It's the amount of CHAT. Okay, so CHAT is the rate limiting step in acetylcholine synthesis. So if I eat more choline, I don't get more acetylcholine necessarily. I need more CHAT. Okay, that's an important concept. Choline, again, is a small chemical. There's no gene for that. Okay, if I want more acetylcholine, I have to make more of the enzyme. These are all monoamines. These three are catecholamines. Serotonin is an indolamine. Okay? Now, if I want to make catecholamines, first I have to eat phenylalanine. That'll get converted into tyrosine. There's an enzyme called tyrosine hydroxylase. I want you to know that. Most of us are going to abbreviate it TH. Okay, so tyrosine hydroxylase is the enzyme that's going to take tyrosine and convert it into dopa. Okay, there's another enzyme. You don't need to worry about the name. This is going to take dopa and convert it into dopamine. Dopamine will then be converted by another enzyme into norepinephrine. Norepinephrine will be converted by yet another enzyme into epinephrine. Okay, so for each enzyme, there's a gene. Okay. So let's say I am a dopamine neuron. Okay. If I'm a dopamine neuron, I want to make dopamine. I'll take phenylalanine, I'll convert that to tyrosine. Will I have tyrosine hydroxylase? Yeah, I need tyrosine hydroxylase to take tyrosine and convert it into dopa. Will I have this enzyme that converts dopa into dopamine? Obviously, yep, I will. Now, what about this enzyme that converts dopamine into norepinephrine? Will I have that enzyme? No. I do not have that enzyme if I'm a dopamine neuron. If I have that enzyme, any dopamine I make will be converted into norepinephrine. That'll make me a norepinephrine neuron. Okay, so if I'm a dopamine neuron, I make all the enzymes up until where I want to stop. Okay, so again, I'll make tyrosine hydroxylase. I'll make this enzyme, but I won't make this enzyme. If I'm a norepinephrine neuron, I'll make this enzyme, I'll make TH, I'll make this enzyme, I'll make this enzyme, but I will not make this enzyme. Okay. Serotonin is derived from tryptophan. Tryptophan will get converted into a precursor, which will then get converted into serotonin. Okay? So out of this chart, I want you to know these metabolic pathways. If I wrote it down like chat, I want you to know that enzyme. I want you to know tyrosine hydroxylase. I've talked about dopamine, norepinephrine. I want you to know the names. You do not have to memorize the structure, but you should know the names. You should also know the classification. So the monoamines, again, there's 4 you need to know. Dopamine, norepinephrine, epinephrine, and serotonin. Now, I mentioned neuropeptides versus neurotransmitter. There's a few key differences. Neuropeptides tend to have longer activity, and that activity is going to be spread out over a wider area. Okay, so neurotransmitters are really fast. Neuropeptides are kind of slower. Think about neuropeptides as sort of modifying long-term behavior. So if you look at this chart, again, you can see a comparison of neuropeptides and other neurotransmitters. Neuropeptides are primarily made in the cell body. Neurotransmitters are made in the presynaptic terminal. The neuropeptides are released primarily from dendrites, also the cell body, and sometimes the axon. Other neurotransmitters are primarily released from the axon terminal. A single action potential causes a release of neurotransmitter. So you know, hopefully remember, the voltage-gated calcium channels open up. Calcium comes in, causes the vesicles to fuse, neurotransmitter to be released into the cleft. Okay, that's going to be other neurotransmitters. Neuropeptides, though, one action potential won't do it. We need repeated depolarization, and that's going to cause the release from the cell body. Neuropeptides are going to diffuse to a wide area and have an effect over many minutes. Neurotransmitters are primarily going to affect receptors on that postsynaptic neuron, and this lasts, has a fairly short effect, less than a second to a few seconds at most. Okay, so neuropeptides are going to be more modulatory. When we get the units on feeding behavior, sexual behavior, you'll see neuropeptides play a bigger role. This is a cartoon of a synaptic vesicle. A synaptic vesicle, remember, is a membrane sphere containing neurotransmitter. When I draw it like this, it looks pretty simple. But in fact, there's many different proteins on it. All these proteins are going to be very important for regulating the release of neurotransmitter. If there's mutations in these, then we won't be able to release neurotransmitter normally, and that can lead to all kinds of problems. There's drugs that target these that absolutely disrupt neurotransmitter release. In one of our cases, you're going to learn about SNAP25, so we'll talk about that more later when we get to the neuromuscular junction. The point of this is that when I say calcium comes into the cell, calcium interacts with the vesicle and causes release of neurotransmitter, I just want you to realize that that's a very complex process. There's literally textbooks just on that process. Now, what are the steps? Again, here's more detail. We've got our action potential coming down. That's going to cause voltage-gated calcium channels to open up. Calcium's going to enter. When calcium comes in, it's essentially going to allow this vesicle to dock to the membrane. When this docks to the membrane, when it's close, then it's going to fuse with the membrane. When it fuses, we end up getting the release of neurotransmitter into the synaptic cleft. Okay, so again, this is just a little more detail about how it occurs. What would happen if I blocked those calcium channels? Well, if I blocked the calcium channels, calcium wouldn't be able to come into the cell. Neurotransmitter would never be released. What were to happen if I were to just inject a whole bunch of calcium here? If I put a whole bunch of calcium in that case, that would cause this docking to occur without an action potential. Okay. So those are the basic properties of neurotransmitters, the real basics of neurotransmitter release. Once that neurotransmitter is released, then it's going to have to move across the cleft and interact with receptors on the postsynaptic cell. In the next video, I'll spend some time talking about postsynaptic receptors. Thank you.